Wow. So lovely to see so many faces in the flesh and so many of you online as well. It's one thing giving a talk in front of your peers and your colleagues. It's quite another doing it in front of your kids who are already starting to snigger in the back of the lecture theatre. So yeah, I can see you. So I thought I would just start by giving a few personal reflections on my journey, in particular, why I'm so interested in epidemiology as a subject. Now, as some of you know, not all of you, some of you, I am an identical twin, and Bex is here tonight. And wonderful as it is being an identical twin, one of the slightly irritating things about it is that your traits are constantly being compared, especially when you look as alike as we do. And what I always remember being a kid is being constantly perplexed as to why it was that Bex had quite severe asthma in her childhood and I didn't have any problems at all. Why was it I always had to trog home on the bike to pick up her Ventolin that she'd forgotten yet again, and I had no problems at all with breathing? And so I became really curious as to why she had a health condition and I didn't at all, when we were so similar in so many other respects. And so that made me really curious as to why some people have different health outcomes compared to others. But of course, relying on individual stories is pretty useless. You need large-scale studies in the general population in order to really find out why people develop some diseases and other people don't. So my journey, like most people, was pretty wandering and fairly aimless, maybe a bit more aimless than most, before I came to Oxford in 1997 to do my DPhil with Tim Key, who I know is here in the audience, who was a fantastic supervisor and has been a great mentor to me ever since. And the two take-home messages I took away from my DPhil here was, first of all, you need really large numbers in order to detect small effects of exposures on disease outcomes. But secondly, it was the secret power of blood samples. So up until then, I had absolutely no idea that you could measure hormones, metabolites, and proteins in the blood that could start to unlock our understanding of the mechanisms through which lifestyle and genetic factors could influence disease risk. And this was a real eye-opener for me. And this molecular side of epidemiology is something that I've been really interested in from a research point of view ever since. So my DPhil focused on trying to find out the dietary and genetic determinants of hormone levels in men and how that might be related to subsequent risk of prostate cancer. So the main finding from my DPhil was that circulating levels of the growth factor called insulin-like growth factor 1, or IGF-1, was higher in meat eaters than it was in vegans, and seemed to be particularly related to a higher intake of animal protein. Now, IGF-1 is interesting because it's a growth factor that circulates in the blood that increases the growth of cells, including prostate cells. And there had been some evidence from other prospective studies showing that men with relatively high levels of IGF-1 may be related to an increased risk of prostate cancer. So what we wanted to do was to really find out whether this was true. Do men with high IGF-1 have an increased risk of prostate cancer? So that led Tim and I to set up the Endogenous Hormones, Nutritional Biomarkers and Prostate Cancer Collaborative Group. Doesn't exactly roll off the tongue. I know, Tim, we really ought to find a snappier name for this. It's a collaborative group to determine finally whether IGF and other growth factors and hormones are related to increased risk of prostate cancer. So the collaborative group, run by myself and Tim and Ruth and Aurora, who are here, the aim of it is to bring together all of the worldwide prospective data into one place so that we can perform a unified analysis to better understand the association between circulating hormones, growth factors, and nutrient biomarkers, and how they're related with the subsequent risk of prostate cancer. So there are about 40 prospective studies from around the world. Thousands of prostate cancer cases and controls are included, with a wide variety of biomarkers. So I just want to show you a few highlights from this very successful collaborative group. So one of the first outstanding questions he wanted to answer was, is circulating IGF-1 level associated with an increased risk of prostate cancer? So when you pull together all of the worldwide data, what we found was that increasing levels of IGF-1 are indeed related to an increased risk of prostate cancer, with men in the highest fifth of circulating concentration having about a 29% increased risk of developing prostate cancer. Now, this was a really exciting finding because IGF-1 is the only modifiable risk factor so far identified for prostate cancer. So the other risk factors, such as increasing age, family history of the disease, being of ethnic minority, there's nothing you can do about those factors. Whereas our previous work had shown that diet could modify circulating levels of IGF-1, particularly a diet in high animal protein, and in turn, high levels of IGF-1 are associated with an increased risk of prostate cancer. The other outstanding question we really wanted to ask was whether circulating testosterone is associated with prostate cancer risk. Now, this has been looked at in a lot of studies previously, but they've been pretty small and have shown inconsistent findings. And the prevailing hypothesis was that high circulating testosterone must be related to an increased risk because the mainstay of treatment for the disease is hormone therapy, which seeks to drastically reduce the man's circulating level of testosterone, which in turn shrinks the prostate and the tumours within the prostate. However, what we found, when you bring together all of the worldwide data from 20 prospective studies, 7,000 cases, 12,000 controls, you can see that there's no real evidence for increase in testosterone in relation to an increased risk of testosterone. However, when we looked at circulating free testosterone, we find a slightly different story. So most of this testosterone that circulates in a man's blood is bound to binding globulins, which means it's unable to attach itself to the androgen receptors and have any biological effect. Only about 2% of circulating testosterone circulates in the blood in its free form, so it's able to attach to the androgen receptor and have a biological effect. So when you measured this biomarker, what we found was that it was men in the very lowest tenth of the distribution that had a significantly reduced risk of developing the disease. And of course, you can only find an association like this when you have such large numbers that you can look at the association in the really extreme ends of the distribution. When you group those categories of tenths into categories of fifths, you can see the association even more clearly. So what do we think might be going on here? So our working hypothesis is that actually at fairly low levels of circulating free testosterone, the androgen receptors are already saturated, so that any further increase in testosterone doesn't lead to any further increased risk of prostate cancer. It's only when you have very low levels of circulating free testosterone that leads to reduced androgen receptor signaling and therefore reduced growth of prostate cancer cells. We did investigate whether this association might be due to bias, in that men with low free testosterone also tend to have low PSA. And of course, PSA test is the first test that men have in order to determine whether to have a biopsy to see if prostate tumours are present. So if you have very low free testosterone and a low PSA test, you're much less likely to have a biopsy and therefore be diagnosed with the disease. So the beauty of having all of the worldwide data performed across such a long period of time was that we were able to look at this association in studies that were performed before the widespread introduction of PSA testing and those studies that were done after the introduction of PSA testing. And we actually found no real difference in this association, which suggests to us at least that this reflects a real biological phenomenon rather than any form of detection bias. So it's been an absolute joy to work on this collaborative group with the Cancer Epidemiology Unit and all of our collaborators worldwide, because it really shows you that you really need very large numbers in order to robustly answer these outstanding questions of biomarkers and hormones in relation to prostate cancer risk. And it means that you can look at these associations to see how they differ by age at diagnosis, ethnicity, disease aggressiveness, and so on. So it continues to provide really fruitful research. So at the end of 2011, I also started to work with UKBiobank. And the main reason for this was I wanted something that was a bit faster paced. Academic research is great, but it took myself and Valerie, it took us 7 years to publish one particular paper. And I'm sure that must be a record somewhere. And so in addition to being, no, not a record. Oh, well, I'm glad I'm not the only one.
It's one of the quickest.
Slaved away. So I wanted something a little bit faster paced, and I also, wanted some different challenges. And little did I know just how fast paced and how many challenges there would be. Rory, I think if we'd have sat down 10 years ago and you'd have said, "Now, this is all the things that UKBiobank will achieve over the next 10 years. Honestly, I think I would have laughed you out of the room. Really. It's been an astonishing ride working with the UKBiobank project, and it's been such a privilege to work with all of you who work with us on this, and we've achieved so much. And now UKBiobank is arguably the best biomedical resource for health-related research. So for friends and family in the audience who perhaps don't quite know the ins and outs of UKBiobank, although I know I do talk about it quite a lot, UKBiobank is a study where we follow the health of 500,000 people in the UK to enable researchers worldwide to better understand how lifestyle, genetic and environmental factors cause diseases in some people, but not in others. It's arguably the most accessible, in-depth, largest, easy to use and flexible biomedical data resource in the world. And our underlying mission is to enable the global research community to make scientific discoveries to improve human health. Now, I know that seems really grandiose and perhaps a bit over the top, but actually, over the last few years, I'd say that's exactly what UKBiobank's been doing, and it will continue to do over the next 10, 20 years. So why is UKBiobank so unique? Well, it's a combination of factors. So we have very large-scale data on 500,000 participants, aged 40 to 69, when they were recruited about 15 years ago across these different assessment centres you can see there on the map. We've collected an awful lot of information about these participants, lifestyle factors, environmental measures, a whole range of physical measures were performed on them, very detailed genomic data, biomarkers on the blood samples, and in large subsets of the cohort, multimodal imaging and physical activity monitors, and so on and so on. And we also have a linkage of all participants to follow their health through linkage to electronic health records, and we now have about 15 years of follow-up. But perhaps what makes it so unique is that from the outset, UKBiobank was designed to be readily accessible to researchers worldwide, whether from academic or commercial institutions for health-related research. And at the moment, there are about 25,000 registered researchers using the data, 80% of whom are international, and there are already over 4,500 publications using the UKBiobank data, 1,600 of which were published last year alone. So the use of the resource, and therefore the impact that the resource is having on health-related research, is growing at an exponential rate, and just will continue to do so over the next few years, I'm sure. So I don't want to dwell on this too much, but it really is worth just commenting on just how revolutionary having large-scale genetic data integrated into a prospective study like UKBiobank has been on health research. So the release of genotyping data for all 500,000 participants was performed in 2017. We released these data to the global research community, and that led to an explosion of analyses and publications identifying genetic variants in relation to a whole range of different diseases. And that also led to the conception of polygenic risk scores. So if you look at the graph in the middle, this is taken from researchers at the Broad Institute a few years ago, where they created a polygenic risk score for coronary artery disease. And what you can see here is they took all of the genetic variants across the genome that are associated with an increased risk of heart disease, each of which on their own have a very small increase in risk, but taken together, you can see that those individuals in the top 5th percentile of polygenic risk for coronary heart disease have over a 3-fold increased risk of developing the disease compared to the general population. And this is actually equivalent to many monogenic disorders, and their genetic predisposition is independent of known lifestyle factors. So while you can imagine that the use of the polygenic risk scores, how that can be used in personalized medicine. So if you take a scenario where you go to your GP, have a blood sample taken, they measure your genetic profile. If you're unlucky enough to have a high genetic predisposition to developing heart disease, then your doctor may then want to target preventative measures, such as you might want to take statins to reduce your level of cholesterol or modify your lifestyle in order to reduce your absolute risk of developing the disease. So while the clinical utility of polygenic risk scores has yet to be established, very recently the NHS has just started to pilot exactly this. So can we implement genetic testing into primary care to identify individuals who are at high genetic risk of developing heart disease for targeted preventative measures? And this type of work has only really come about through the use of the UKBiobank resource. Now, of course, there's much we still don't understand about genetic variation in the coding and the non-coding regions of genes on health. And that's led to a consortium of big pharmaceutical companies coming together to invest millions of pounds in UKBiobank to perform whole exome sequencing, which measures the 2% of the genome that encodes for proteins, and whole genome sequencing, which measures all the genetic variation across the genome. And these data will dramatically enhance the ability of UKBiobank to support really innovative research into the genetic determinants of disease. But of course, this has only been made possible by huge advances in technology. And it was only in 2003 that the data was published from the Human Genome Project that released the whole genome sequencing for the human. And it took over 10 years to perform and over $2 billion to do. And here we are now performing whole genome sequencing on 500,000 participants in the space of about 3 years. I mean, it's astonishing the speed of technology change that is therefore leading to huge potential to increase our understanding of disease determinants, and also to increase the likelihood of successful drug discovery targets by identifying functional variants that can then be related to changes in drugs to improve disease outcomes. So it's hugely exciting that UKBiobank's been able to perform all of this detailed genomic work incorporated into a prospective resource. But of course, UKBiobank is much more than a collection of biological samples for health-related research. In addition to its large size, the phenotyping, the genetic data, it's a prospective resource, so we follow up people over many decades to find out what happens to their health. And this is something I want to talk about in a little bit more detail, as it's something that the group here in Oxford are largely responsible for. So in order to do this, we need comprehensive, scalable and cost-effective methods to link to longitudinal health records. So the data analyst team here in Oxford, led by Ed Horn, have done a huge amount of work in following the health of 500,000 participants by linking to national death and cancer registries, hospital inpatient admissions, and for just under half of the cohort, we have primary care data up until 2017. I'll talk a bit more about that in a moment. Now, at the start of the pandemic, we really wanted to make all of these healthcare records available regularly and rapidly to researchers to enable COVID-19 research. So we were able to obtain primary care data through emergency legislation to UKBiobank for the sole purposes of COVID-19 research. We were also able to link to the PCR diagnostic antigen test since the start of the pandemic, and also to critical care records. And all of these data sources were made available on a regular basis, approximately every month, to researchers to facilitate COVID research. All told, there are well over 250 odd papers published looking at the genetic determinants of severe COVID-19, how circulating metabolites are related to disease, how underlying comorbidities and treatments are related to an increased risk of COVID-19. And all of that work has been enabled because of these rapid linkages that we have for all participants. Now, linkage is not as easy as it sounds. There are different data providers in each of the 3 devolved nations. Our participants are from England, Wales, and Scotland. There are substantial administrative and governance hurdles to overcome in each of the 3 devolved nations, and Gretchen in our team is becoming. So Gretchen knows exactly what I'm talking about in terms of governance hurdles to get our hands on the data. And then when we do receive the data, there's different variables that mean slightly different things, different coding schema across the different data sets. We have a huge volume of data we have to process. There's up to 100,000 hospital admissions each year. And there's varying accuracy of the diagnostic and procedural codes across all the different data providers. And in terms of the GP data, so as I've said before, we were able to obtain GP data for just under half the cohort up until about 2017, at which point we were kind of stymied. And even now, GP data is still not available for general research, despite the fact that we have explicit consent from all of our participants to link to their health-related records. Now, there are various reasons for this. There's no single source of GP data. It exists in electronic system suppliers, of which there are several across the UK. The data is controlled by individual GP practices, and they're often too busy, unwilling, or perhaps unsure of the data sharing rules to provide approval for the UKBiobank data for their patients to be transferred to us for health research purposes. So we tried various methods to encourage GPs to press the button on their screen to transfer the data, and despite all our best incentives, only about 20% of GPs do so. So it's obviously not a constructive way forward. So there's hope, there's silver linings in all this. In that hopefully in the next year or so, there will be a centralised GP data set from NHS Digital for general research purposes. So we wait with bated breath for that. But the reason why GP data is just so important for studies like UKBiobank is because it increases the numbers and the range of health outcomes that can be detected. So here's just a handful of conditions, and you can see the numbers in black text are the numbers of incident cases by the end of 2020 occurring in UKBiobank if we just have a linkage to hospital and death data. And the numbers in red are those additional number of cases that we would have if we were able to access GP data. So you can see that if you're a researcher interested in myocardial infarction, having access to GP data would actually only increase case numbers by about 6%. However, if you're interested in conditions like diabetes, COPD, or Alzheimer's disease, that increases case identification by between 40 and 50%. And if you're interested in conditions like rheumatoid arthritis, anxiety, asthma, these conditions are predominantly only ascertained in primary care. So that's why having access to these data is just so critical for a resource like UKBiobank to enable research into these common but nonetheless debilitating conditions that at the moment have been under-researched precisely because we don't have access to GP data. Okay, so in addition to the record linkage, the group here in Oxford, we're also responsible for linking to asking participants to ask about various health exposures that we didn't have enough time to ask them at recruitment, so things like diet and occupational history, but also about health outcomes that are difficult to assess through linkage. So things like cognitive function, mental health, gastrointestinal symptoms, and pain. Now, the work on questionnaires was stalled for a bit during COVID as we were busy doing other things. We just finished a cognitive function repeat questionnaire to enable research to look at changes in cognitive function over time. And over the next 18 months or so, we plan to administer questionnaires on health and well-being with a focus on long COVID symptoms, as well as sleep, autistic traits, and a repeat of the mental health questionnaire to enable research into these kind of more episodic or progressive conditions. This work is coordinated by Joe Holliday and Fen Starkey in our team who do a huge amount of work in talking with the clinical experts, coming up with valid questionnaires that are nonetheless simple, quick, and acceptable for our participants to use. Now, most of these questionnaires are available for use in other research studies via the Oxford Questionnaire Sharing Service, which is largely built by Alan and coordinated by Jill Rodd in our team. And the whole point here was to try and minimize duplication of effort by other studies who want to use our questionnaires in order to harmonize data for pooled analyses. So I've talked a bit about how much advancements in exposure measurements, and particularly related to genomics, has really improved research into the genetic determinants of disease. And now the same attention needs to be applied in how we measure health outcomes. There are currently no standards for how we select and combine different codes to define a disease, which means there's substantial variability in the way that different researchers define the same disease. So what we need are different approaches to combine the data across all the diverse healthcare records and questionnaires that we have to characterize the off-the-shelf outcomes that are both valid, comprehensible, and scalable to all the health outcomes that exist in the database. This schematic is a nice way of showing the different type of approaches that exist. It's taken from John Robinson's paper a few years ago. Now, up until quite recently, UKBiobank, we've been focused on these orange and blue blobs down here, based on fairly high clinical effort, but very low informatics expertise needed, where we've generated health outcomes using algorithms based on Boolean logic, based on clinically code lists that are debated amongst clinicians and then verified in an iterative process using manual review against the full clinical record in a small subset of the cohort. This has turned out to be really laborious, time-consuming, and it's just simply not scalable to do this for all the health outcomes that exist in UKBiobank. So we're also thinking about, well, can we use machine learning approaches, which is these yellow and green blobs up here, in order to classify participants with a certain health outcome based on the pattern of disease and procedural codes that exist in their health records. Obviously, this requires a lot of informatics expertise, and so we plan to reach out to the research community through a series of data challenges to see if we can use these type of AI-type approaches to help us better phenotype health outcomes across the diverse set of healthcare records. In the meantime, we're working on this grey blob down here, not because it's the least amount of clinical and informatics effort, although that might be part of the reason, but actually because it's what researchers are asking for. So what we've been doing is mapping all the different codes that exist across the different data sources that we have to a single coding schema using the ICD-10 three-digit classification, which is the most commonly used classification for health outcomes. And so we've created these first occurrence fields in the data showcase, which essentially gives you the date of the first occurrence of codes that are mapped to an ICD-10 code in the UKBiobank. So this is an example for depressive episode. You can see there are about 58,000 participants with this particular code coming from self-report at recruitment, hospital admissions, and primary care. So while this is sufficient for broad brush research into whether someone's had a depressive episode or not, if we were able to map to a 4-digit ICD-10 code rather than a 3-digit code, it would enable researchers to distinguish between the severity of the condition. So, and this is work that Ben Lacey and Chi Feng and the epidemiology team are working hard to assess the feasibility of whether we can map to these more granular levels of outcomes. So creating these kind of off-the-shelf codes and phenotypes that are mapped to ICD will hugely facilitate research into a wide range of health outcomes because it will simply minimize the duplication of effort that researchers are already doing. So I just want to spend the last 10 minutes or so talking about the work that we've been doing for COVID-19 research. So the last 2 years has been pretty exceptional, I would say. The team has worked their absolute socks off over the last 2 years to set up several studies to enable the research community to perform research into COVID-19. And the fact that we already have a large prospective study with a high degree of participant engagement means that we were able to quickly mobilize our resources to enable COVID research. So very early on in the pandemic, the Department of Health and Social Care came to us and asked if we could set up a study to determine the extent of past infection in about 20,000 individuals across the UK to see how it varied in different population subgroups, what are the most common symptoms, and perhaps most importantly, to look at antibody persistence over time. It was one of the few studies in the UK that was being set up then in order to see how long antibodies persist after infection. It was a huge team effort across the whole organisation. I think most of us slept, ate, drank the study for about 10 months. It was a lot of effort to get the study off the ground. Amazingly, about 120,000 people signed up to join the study. We were overwhelmed with the response. And that enabled us to recruit about 10,000 UKBiobank participants and 10,000 of their adult children and grandchildren in order to increase representation across age. And it was the first time we'd ever asked the kids to get involved in the research. And we've been really amazed at just how committed they have been to sticking with this study. Because we had so many people interested in joining the study, we were able to stratify recruitment across different age ranges, by sex, regions of the UK, and we were also able to oversample those of ethnic minorities, those who lived in deprived areas, and those who lived in London, because we knew that rates of infection were highest in those particular groups. So we mailed this blood sampling kit and a questionnaire symptom survey that we devised to all 20,000 participants every month for 6 months between June to November of 2020. We can see here pictures of the Royal Mail delivery van every day delivering samples to our processing labs up in Stockport. The samples were then checked, processed, aliquoted, plated up and couriered down to a lab here in Oxford in order to measure antibodies to the spike protein. So what we found was that the seroprevalence, which is simply the proportion of the population who had positive antibodies to coronavirus infection, increased from just under 7% in June to just under 9% by the end of November of 2020. We saw that seroprevalence varied substantially across regions of the UK, being highest in London and lowest in the southwest of England and in Scotland. It also varies substantially by age, being highest in the youngest age groups, and by ethnic group, being highest in those of Black ethnic minorities compared with the white population. And when we looked at duration of seropositivity, what we found was amongst those who were positive at the start of the study, almost all of them were positive 3 months after the onset of symptoms or the date of their PCR test, and about 88% were still positive 6 months later. Now, when these results were sent to the DHSC in December of 2020, it was one of the few studies that had serial measures on participants to really enable us to really look at this question. And I know this kind of seems obvious now, but at the time, nobody really had a good handle on just how long antibodies lasted following infection. We've now just sent a final sample to all 20,000 individuals in order to assess antibody persistence over a much longer period of time, so 12 months or more. And these data are just flowing in now, and that should be able to, again, be one of the few studies in the world to look at long-term antibody persistence over time. So one of the things that came out of the study was, and other studies actually, was that Black and ethnic minority groups had a higher risk of developing COVID. So what we wanted to do was to enable research to understand why it is that ethnic minorities are higher risk of developing the infection. So we sent out a questionnaire to all 20,000 participants asking about some socio-demographic factors and behaviour during the pandemic. Now, this was important because previous studies had shown that socioeconomic status was important in explaining these ethnic inequalities, but they lacked kind of the detailed data on which specific aspects were relevant. So the data from our survey showed that actually, once you adjust for occupation, such as whether people worked within the NHS, social care, or had a public-facing role, and geographical factors, whereabouts they lived in the UK, whether they lived in an urban or rural area, that reduced their risk by about 2/3. And once you've done that, then additional adjustment for how many people lived in the house, how they got to work, socioeconomic status, and education had actually very little additional impact. So this work was done by Wemimo and Ben and the epidemiology team, and really suggests to us that it's actually the job that you do and whereabouts in the UK that you live that is the strongest drivers of these ethnic inequalities in COVID-19. So last year, we also set up a couple of studies to enable research into the longer-term health effects of coronavirus infection. So we were funded by the Department of Health and Social Care to send out home-based antibody lateral flow kits to as many participants as possible in order to obtain objective evidence of past infection. And the kits were kindly dispatched free of charge by Amazon. Now, this study was done between March and June of last year, and I can't tell you the frustrations that we had in the delays in obtaining MHRA approval to use these two different types of lateral flow tests in the home for health-related research. The delays took so long to get MHRA approval, by which time people were having their first vaccination. And the problem for us is that these lateral flow tests didn't distinguish between antibodies produced by a vaccine and antibodies produced by infection. So it really was a race against time. So by the time we did get MHRA approval and the study was done, we sent out these home-based tests and we got results back from about over 200,000 participants, about 64,000 people who had already had their first jab. So then we had to then send them another sample collection kit to do a small amount of blood into a test tube. That got sent off to a different lab to determine whether the antibodies were from vaccination or were from infection. At the end of all of that, about 6% of the study population had evidence of previous infection of coronavirus by the middle of last year. So what will these data enable researchers to do? So now we have objective evidence of previous infection with coronavirus. These data, combined with the longitudinal health records we have and the questionnaire data that we have, will enable researchers to look at the longer-term health effects of infection across the full disease spectrum. So some of these individuals had no idea they had COVID, they were completely asymptomatic, up into people who were hospitalized with the disease. So finally, last year, we also were involved in helping to set up a coronavirus imaging study. Quite early on in the pandemic, there was some evidence coming out that coronavirus is not just a respiratory infection, and that it might be associated with multi-organ injury that may have medium to long-term consequences. So our aim was to generate a unique data set to assess the effect of infection on changes in internal organs by performing imaging scans on people before and after infection. Now, this was really crucial because all of the studies that have been done looking at imaging in relation to COVID have recruited individuals after they developed the infection, and they've usually recruited individuals who've been hospitalized with the disease. So we were able to do a study that collected data before and after people had had the infection and across the full disease spectrum. In order to do this, we took advantage of our ongoing imaging study that aims to perform MRI scans on up to 100,000 individuals. So 50,000 participants had already attended an imaging assessment before the pandemic. So we invited 2,000 of them, half of whom had previously had COVID, either through evidence from their lateral flow test or through linkage to their PCR test data, half of whom didn't. We invited them back for a second imaging scan last year. And it's the only study in the world with both pre and post-infection imaging data. These data were released to researchers as and when they were being collected, and research is already starting to be published showing that participants with previous infection have a greater reduction in grey matter thickness in the olfactory region of the brain compared with those who have not been infected. And may also be associated with a greater reduction in cognitive function, at least over the medium term. So it's already producing really novel and interesting findings. So finally, those of us who work on UKBiobank data we're used to dealing with lots of data, millions and millions of numbers every day. And it's easy to forget that we're actually tracking the lives and the deaths of real people. So this is a photo of my dad, who's here tonight, and his wife, Deb, both of whom are UKBiobank participants. Now, Deb was diagnosed with an aggressive form of brain tumour a couple of years after she was recruited into the study. And whilst she survived for many years beyond the doctors predicted, unfortunately for us, she died a few years ago. And I'd like to think, actually, that UKBiobank is probably the only study in the world right now that can reliably answer the question as to why it is that people like Deb are more likely to suffer these types of brain tumours, and why it is that people like Deb are more likely to survive compared with other people of her age. Now, UKBiobank, we've achieved astonishing things over the last 10 years, and I think it is true to say that we're now, one of the world's largest and most used and most available biomedical research for health-related research, and will continue to do so for the next 10 or 20 years. But actually, it's the altruism and the ongoing commitment of all 500,000 participants that is really the most astonishing. So finally, I just want to thank a huge thank you to all of the UKBiobank team here in NDPH. I can't thank all of you individually, but it's been an absolute privilege to work with you over the past few years on this remarkable journey. I also want to thank the support teams here in NDPH, the IT teams, the programming teams, the testing teams, who often don't get a mention, but your work is crucial for us to be able to do our day job. And also, particularly, I do want to thank Rory, as well as being fantastic leader of the department, as leader of UKBiobank. I think it's been your vision, in your single-minded determination and work ethic that's made the UKBiobank study what it is today. I'd also just like to thank family and friends who are here tonight, who've really supported me over the years. You've travelled from Birmingham, from Brighton, from Devon to be here to support me, so thanks for that. And finally, I just want to thank Aid, my husband, and Zoe and Lizzie for showing me actually what the priorities are in life. Between the three of you, putting dinner on the table every night. And for that, I am truly, truly grateful. So thank you very much.